Welcome to this lecture on the principle of immunology. Today, we are going to explore one of the most fascinating and powerful defense mechanisms in the human body, the complement activation pathways. If you have ever wondered how our immune system can instantly recognize and overwhelm a bacterial invasion before our white blood cells even arrive on the scene, you are in the right place. We will build our understanding from the ground up, looking at both the biology and the mathematics behind this incredible system. Let us start with a simple analogy. Imagine a medieval castle under attack by a stealthy enemy in the dead of night. The guards on the wall do not just fight the invaders one-on-one. Instead, they light a massive signal fire. This first fire instantly ignites a second, which ignites a third, until the entire kingdom is awake and a massive boulder is dropped on the invaders. In our bodies, this brilliant signal fire system is called the complement system. It is absolutely vital because it allows a tiny, localized threat to trigger a massive, overwhelming, and immediate defensive response across the body. Now that we have our analogy, let us look at what this system actually is, biologically. The complement system is a complex network of over 30 soluble plasma proteins and membrane-bound receptors. It functions as a major effector mechanism, bridging both our innate and adaptive immunity. These proteins are mostly synthesized in the liver and circulate quietly in your blood as inactive precursors, which scientists call zymogens. They are essentially floating around, waiting for a specific trigger to start an unstoppable chain reaction to protect you from harm. So, how do these inactive proteins actually wake up? When stimulated by specific triggers, such as a bacterial surface or an antibody, these zymogens are cleaved by enzymes into active fragments. Here is where the magic happens. These newly activated fragments then act as enzymes themselves to cleave the next protein in the sequence, creating a massive cascade. Ultimately, this sequential activation converges on a central critical step, which is the cleavage of a protein called C3. This specific cleavage event is the turning point that leads directly to the destruction of the pathogen. Before we dive into the mathematics of this cascade, we must understand why this intricate system matters so much to our survival. Without the complement system, our immune cells would be virtually blind and far too slow to respond to rapidly dividing bacteria. It acts as the crucial, lightning-fast bridge between the innate and adaptive immune systems. In real life, individuals with genetic deficiencies in complement proteins suffer from recurrent, life-threatening bacterial infections. Furthermore, understanding this system allows doctors to develop targeted therapies for severe autoimmune diseases, where the complement system mistakenly attacks the body's own healthy tissues. To truly appreciate the explosive power of the complement system, we must look at the mathematics of the alternative pathway's amplification loop. Once a single C3b molecule binds to a pathogen, it forms an enzyme that creates even more C3b, resulting in a positive feedback loop. We model this using a standard exponential growth formula. N of t equals N0 times e to the power of kt. Here, N of t is the total number of C3b molecules at a given time. N0 is the initial number of molecules. The constant E perfectly models continuous growth. The variable K is the amplification rate constant, and T is the time in seconds. If the rate constant K increases, the cascade happens much faster, leading to a more rapid destruction of the invading pathogen. Now that we have our formula, let us see this with a concrete example. Let us imagine a scenario where a single C3b molecule spontaneously binds to an invading bacterium. We are given an amplification rate constant of 0.5 per second, and we want to find the total molecules after exactly 10 seconds. We substitute our known values into the exponential growth model. N0 becomes 1, K becomes 0.5, and T becomes 10. Multiplying the rate constant by the time gives us exactly 5 in the exponent. Evaluating e raised to the 5th power gives us approximately 148.4. Since we cannot have a fraction of a molecule, our final answer is 148 complete molecules. Physically, this means in a mere 10 seconds, the bacterium is heavily coated with molecular eat-me signals, preventing its escape. This rapid tagging we just calculated leads us to the final, lethal stage of the complement cascade. This stage is the formation of the membrane attack complex, commonly referred to as MAC. While C3b tags the pathogen for destruction by other cells, the MAC takes matters into its own hands. It acts as a direct, lethal weapon by punching physical pores into the bacterial surface. When enough of these pores are formed, the bacterial cell can't maintain its internal pressure, causing it to lyse or burst open. This highlights the incredible efficiency of the complement system in clearing infections rapidly, completely independent of other immune cells. To understand how quickly this destruction happens, we use a mathematical function that models the accelerating rate of MAC pore formation. Laboratory observations for a specific bacterial strain give us the formula m of t equals 100 times t squared. In this equation, m of t represents the total number of MAC pores formed on the bacterial surface, and t is the time measured in minutes. Notice that this is a quadratic relationship, meaning the rate of pore formation accelerates as time goes on. To successfully burst open this specific bacterial cell, a critical lethal threshold of at least 5,000 MAC pores must be reached. If the coefficient increases, the pores form faster, reducing the time the bacteria can survive. Let us apply this formula to determine exactly how long it will take for the complement system to completely destroy the cell. Our strategy is to find the exact moment when the number of MAC pores reaches the critical threshold. We set our function, 100 times t squared, equal to the required 5,000 pores. To isolate the time variable, we divide both sides by 100, which leaves us with t squared equals 50. Finally, we take the square root of both sides. Since time must be positive, we ignore the negative root. The square root of 50 evaluates to approximately 7.07. Physically, this means it takes just over 7 minutes for the complement cascade to punch enough holes in the bacterial membrane to cause it to burst and die. Now that we have calculated the speed and power of this system, let us look at the complete picture. In this diagram, you can see the 3 distinct routes of activation at the top. On the left, the classical pathway is triggered by Y-shaped antibodies. In the middle, the lectin pathway is triggered by specific sugars. On the right, the alternative pathway begins with spontaneous binding. Notice how all 3 of these distinct pathways point downwards and converge on one central enzyme, the C3 convertase. This is the critical bottleneck. From here, the cascade splits into a devastating 3-pronged attack. The small C3a fragment floats away to trigger inflammation, calling for cellular backup. The larger C3b fragment coats the pathogen for opsonization, tagging it for destruction. Finally, at the bottom right, you see the membrane attack complex, forming physical pores in the cell membrane, leading directly to cell lysis. We have covered a lot of ground today, so let us review the key takeaways. The complement system is a vital, rapid response cascade of proteins that tirelessly defends your body against invading pathogens. Whether triggered by antibodies, sugars, or spontaneous binding, all three pathways brilliantly converge to amplify the signal via the C3 protein. This results in a devastating three-pronged attack, tagging the enemy, calling for cellular backup, and directly blowing up the invader with MAC pores. As we saw through our mathematical models, this exponential amplification allows the body to neutralize threats in a matter of minutes. Mastering this concept helps you deeply understand how our bodies naturally fight infection. Great job following along, and thank you for watching.